This video covers false prediction number 2. In biological cells, there are certain fundamental processes and molecules, such as the DNA code, DNA replication, and so forth. The idea here is that these are difficult to change. They're difficult to evolve, so they should be conserved. So, false prediction number 2 is that the cell's fundamental processes and molecules are universal. Let's have a look. Before we look at this false prediction, I want to show you briefly how DNA replication works. Everyone's heard of the DNA double helix. What you may not know is that the two different threads are anti-parallel. They go in opposite directions, and I'll show that to you. Here's an example of two nucleotide pairs in the DNA double helix. On the right, you have adenine and cytosine nucleotides. On the left, you have thymine and guanine nucleotides paired up with each other. On the backbone, the thread part, the part that makes up the threads, you have a phosphate group there at the top connected to a sugar, and then another phosphate group connected to another sugar. So that's going to be the sequence as you go along a thread. The backbone of the DNA double helices are phosphate group, sugar, etc. On the right-hand side, it's the same, except it's going in the opposite direction. You go to the bottom, it's a phosphate group, sugar. I'm going into these details just to get you that idea that they go in the opposite direction, and that's going to be very important to understand DNA replication. We also have this nomenclature of 5' and 3' ends that comes out of organic chemistry. Okay, so the DNA double helix strands are anti-parallel. So what about DNA replication? There's a molecular machine, a big, complicated molecular machine called DNA polymerase, make many DNA. So another machine called helicase that divides the DNA double helix, and then the DNA polymerase slides along one of the threads, one of the strands, and attaches the complementary nucleotides. So they are doing that on both of those strands that have been separated, creating two new double helices. When you have cell division, when a cell needs to divide, it needs to copy everything in the cell, including the genome, the DNA. And so this is what happens with all of the DNA in the cell getting copied like this. But notice that these DNA polymerase machines are going in opposite directions. You see the blue arrows there. So there's a leading strand and a lagging strand. And the leading strand, the DNA polymerase just simply goes along continually toward the fork, toward the helicase that's dividing, splitting the double helix. Interestingly, on the lagging strand, it's going in the opposite direction. Because these are anti-parallel, the DNA polymerase needs to work in the same direction, so to speak. So it goes in opposite directions in these two different strands. It needs to go in the same direction relative to the chemistry of the strand. Here's another illustration of that. So it's going to be kind of, it makes sense on the leading strand that as you separate the double helix, the DNA polymerase simply continuously moves forward, copying, grabbing nucleotides and attaching them to the strand. So it doesn't have to do anything, too clever. It's very clever, but it doesn't leave the strand. It just continuously creates that template, that second strand. On the lagging strand, something much more complicated happens. And to do that, what I want to do is switch to an animation from Fun Biology, and there's the link to it. I recommend Fun Biology. Go check it out for neat videos like this one. But let's have a look so you get an idea of how this DNA replication works, especially on the lagging strand.
In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment.
Okay, so that gives you an idea of how DNA replication works, and especially that incredible complexity on the lagging strand, and how DNA polymerase has to go along and then dismount and move back and go along some more, and the priming that needs to occur, and all the different actors that have to play their role, so to speak, do their job. Very complicated process. It is also one of the cell's fundamental processes. DNA replication is complicated. Okay, so we got that. So what about this prediction? The cell's fundamental molecules and processes are universal. So the rationale here is that foundations cannot easily change, right? You take a skyscraper, you want to change the antennas on the roof, you want to paint the hallways a different color, change out some windows, fine, you can do all that. You can't go in and change the foundation. And that's really the idea here, is that as evolution rumbles along, you can't change foundational aspects of life, things that DNA replication, DNA code, the fundamental processes in the cell. You can't just go and evolve those. So those need to be universal across all life. This was a strong prediction of evolution, and it was strongly celebrated as well. Evolutionists definitely felt that this had been strongly confirmed. I'll give you some examples here. Niles Eldredge wrote that the underlying chemical uniformity of life was a severe test that evolution passed with flying colors. So underlying chemical uniformity, he's talking about the DNA code, DNA replication, things like that. It was a severe test, very, very significant prediction, and that evolution had successfully passed that. Nobel laureate Christian de Duve, he wrote that evolution is in part confirmed by the fact that all extant living organisms function according to the same principles: DNA code, DNA replication, and so forth. Michael Ruse, a top Darwin scholar, wrote that the essential molecules of life help to make evolution beyond reasonable doubt. So you have these evolutionists claiming victory here, but it was premature. This prediction has been falsified, no question about it. Let's have a look here. So let's first consider the DNA code. We looked at that in false prediction number 1, and we saw this chart in particular, which kind of laid out the process, diagrammed the process of protein synthesis in the cell. So you have these different components, and take a special look at the tRNA molecule there, the yellow box molecules there. They're carrying an amino acid on one end. So they're carriers of amino acids, and they contribute those amino acids to the protein synthesis process. They come along and give up their amino acid, then they go away uncharged. They're empty. They've lost their amino acid. They need to be recharged. So something has to put that amino acid to charge the tRNAs with the correct or the cognate amino acid initially, and then once they get used, they need to be recharged. So what does that? We saw in false prediction number 1 that machine is those machines are called amino acyl tRNA synthetases, or AARSs, amino acyl tRNA synthetases. So all that means is an amino acid and a tRNA are synthesized or combined. That's what these molecules do. These are protein machines that go around attaching, charging these tRNAs, these spent tRNAs. And they are the magic. They're the actors behind the scene that actually enact the DNA code. So they're crucial. These are the proteins that actually do the DNA code. And that DNA code is universal, as we saw. However, these proteins themselves are not. In fact, they strongly contradict evolution's common descent pattern. So, in other words, their function is universal, but they themselves are not universal. If you compare them across different species, they do not fit at all the common descent evolution's common descent expectation, common descent model. This is a massive internal contradiction. You have the DNA code, which necessitates a universal common ancestor of all life, and yet it doesn't work on the very same molecules. The molecules that necessitate the universal common ancestor model, that is, by virtue of the DNA code, their function is to enact the DNA code, these very same molecules contradict common descent. Here is a paper by Carl Woese, Gary Olson, and co-authors reviewing this exact problem, the amino acid tRNA synthetases and how they violate, contradict the common descent model. And they talk all about the contradictions, the weird non-evolutionary patterns that they make up, and how they try to solve those conundrums and contradictions. Gary Olson presented much of these results in his comp bio class, and which I religiously sat in on when I was there at Illinois. And Gary did a great job in that class, and it was fascinating to see these results at the time. And he presented them as, of course, interesting puzzles for evolution that they need to work through. But they, in fact, are false. They falsify this prediction, as we'll see here. The AARSs strongly contradict evolution's common descent pattern. So, reading from the paper, much has been said and written about amino acid tRNA synthetases contradicting established organismal phylogenies, that is, the evolutionary tree. From the above analysis, one can see that in many cases they do so, sometimes in spectacular ways. In at least as many other cases, however, they do not. Well, it's fine that they don't always contradict the model. The problem is they often do, and that is a falsification. You can't have occasional failures that don't count. Things don't float upwards, and if that happened, you wouldn't say, well, that doesn't really matter, the gravity is still true. I mean, that would be a disaster for the theory of gravity. So these are major violations of the common descent and evolution model, in particular because the contradictions are so significant they can't be explained by the kind of the usual, oh yeah, some more mutations happened, something was a little different. And that's what this paper will try and get into is, gee, how can we explain this? It won't be easy. Skipping down, however, in a number of cases, a bacterial synthetase is obviously of the general archaeal genre, but resembles neither the archaeal nor the eukaryotic version specifically. This immediately raises a non-trivial question about the source of the synthetase. So this is just one example of many throughout this paper where they're just running into problem after problem, and how are we going to answer this non-trivial question? So we have a prediction and we have a big problem with the AARSs. Let's look at some other ways that this prediction is falsified. And now we get back to DNA replication. We talked about how in DNA replication, a key player is the DNA polymerase. Well, it turns out the DNA polymerase machine also contradicts evolution's common descent pattern. Here's a paper that says, in particular and counterintuitively, given the central role of DNA in all cells and the mechanistic uniformity of replication, the core enzymes of the replication systems of bacteria and archaea, as well as eukaryotes, are unrelated or extremely distantly related. So these DNA replication machines are not universal either. Here's another paper. It says, it is therefore surprising that the protein sequences of several central components of the DNA replication machinery, above all the principal replicative polymerases, show very little or no sequence similarity between bacteria and archaea eukaryotes. In other words, not universal and contradictory. It's not fitting the expected evolutionary common descent pattern. Furthermore, different DNA replication processes have been discovered. So I showed you, we looked at the video, we kind of looked at the canonical DNA replication process with the leading strand, the lagging strand, and so forth. But actually more modalities of replication have been discovered. It isn't universal. This paper explains, viruses and plasmids in addition possess at least two unique DNA replication systems, namely the protein primed and rolling circle modalities of replication. This unexpected diversity makes the origin and evolution of DNA replication systems a particularly challenging and intriguing problem in evolutionary biology. Yeah, you could say that. It's a falsification of the prediction. Now, the DNA code and DNA replication, yes, they are fundamental to the cell, but they're not the only fundamental processes and molecules in the cell. The cell membrane is also fundamental, and important differences across species have been found in the cell's fundamental plasma membrane and lipid biosynthesis pathways. Let's take a look at this paper. It says, besides, we present evidence that at least two biochemical pathways as important as DNA replication and lipid biosynthesis are not homologous between bacteria and archaea. The identities observed between the proteins involved in those pathways along representatives of these two ancestral domains of life are too low to indicate common genic ancestry. So, long paragraph there. Bottom line, more contradictions to the common descent pattern and to this prediction. And you'll notice the title of the paper there. Is it possible that cells have had more than one origin? We'll look more at that in a minute. So we see a prediction clearly made and clearly falsified. Let's move on to the response then, because again, looking at these predictions, what's important is the denouement, the response, the outcome. What happened? What did it force? What did the theory have to go through? If a false prediction leads to new knowledge and everything falls into place and there's a smoking gun, then great. The false prediction was really not a problem. It was a good thing. So what happened in this case? Well, we have a number of conclusions that evolutionists have come to because of this false prediction. The first one is a massive amount of unobserved and ad hoc horizontal gene transfer. Let's look at some quotes from the Gary Olson, Carl Woese paper. And this is an ad hoc and complex, adds complexity to the theory, and it is an ad hoc explanation, as we'll see here. So let's look at some quotes here. One would expect the horizontal gene transfer profiles of the 20 synthetases all to be similar in some general respect. However, this is clearly not the case. The horizontal gene transfer profiles of the different synthetases can be qualitatively different. So in other words, you've got these different AARSs for the 20 different amino acids. You'd kind of expect them to have a different, what they're proposing is a lot of horizontal gene transfer to explain the ways that they contradict the evolutionary common descent pattern. Oh yeah, so we have to have all this, much more than we would have, that we observe today. So there's kind of a horizontal gene transfer on steroids is what they're proposing. But even that is a problem, as they point out here, that they're having to tailor it for the different synthetases. Skipping down, it is essentially self-evident that other factors being equal, most horizontal gene transfers will be taxonomically local. In other words, if you are having this massive amount of gene transfers between species, it's probably going to happen between similar species, sister allied species. You're probably not going to have a lot of horizontal gene transfer between very distant species, distant in many regards, taxonomically and probably geographically and so forth. But again, that's not the case. As they say, we nevertheless think that the data available at present suggests that horizontal gene transfers of the AARSs are not predominantly local. The horizontal displacements that have occurred deep in the phylogenetic tree involving transfers from one major taxon to another major taxon appear to be unexpectedly prevalent. So another problem with this idea, they're calling upon an enormous amount of horizontal gene transfer, but even that is difficult to make work because they're having to employ it between taxonomically distant organism species. Let's move on to another, skipping down here to another quote from the paper. The fact that horizontal gene transfers, the fact that they appear to dominate the landscape of amino acid tRNA relationships suggests that the dynamic of horizontal gene transfer has not remained constant over the evolutionary course covered by the universal phylogenetic tree. A dynamic horizontal gene transfer that changes over the evolutionary course means that the nature of cells has changed over that course as well. So another just so story they're having to implement, which is changing over time. These horizontal gene transfer events must have changed dramatically over time. So a lot of problems there. Another conclusion they came to was rapid evolution. Let's read this. A more satisfying explanation may lie in the classical evolutionist concept of evolutionary radiations, which again involves the tempo-mode relationship. Three important characteristics: accelerated evolutionary tempo and remarkable and remarkably diverse evolutionary invention. So this is something that comes out of the fossil record that has been hypothesized for years is, all kinds of new forms appear abruptly in the fossil record. I guess that's just the way evolution works. It just accelerates dramatically at times, and all kinds of new innovations just appear on the scene rapidly. I guess that's happening as well with these AARSs and this horizontal gene transfer. It just kind of happens out of the blue real fast, and new innovations occur very quickly. Yeah, let's use that explanation that those paleontologists used years ago. We'll apply the same. We can play that game too and take a page out of their book for the AARSs as well. Another conclusion they had to come to was that many of the AARSs simply had to have evolved late. Skipping down here, we think that the lack of canonical pattern shown by all synthetases in the Gemini group has a similar explanation. That is, that all their taxonomic distributions, like that of GLN-RS, were established relatively late in the evolutionary course, well after the domains themselves had arisen and their major branchings had begun to coalesce. So again, they're playing around with the time now and say, many of these ARSs didn't evolve until later on. So tailoring the explanation to the awkward data. Another conclusion was discontinuous evolution. Reading from the paper, in all this, the suggestion that the evolutionary process is not what it might seem when attention is confined to the genome sequence level, i.e. a more or less continuous progression leading from very simple primitive forms to the modern ones. Rather, evolution may proceed through a series of discontinuous stages, with the quality of the evolving systems changing dramatically from stage to stage. In other words, again, they're saying, you might expect for evolution to work in this kind of linear fashion from less complex to more complex and kind of a continuous progression, but we're kind of dropping that model here. We think that it can just kind of be more discontinuous. They're having to do this because the data are contradicting the theory. What we just went through is a series of just so stories tailoring the details of the theory, adding incredible complexity to the theory, losing all parsimony, violating Occam's razor, and trying to explain what they're observing in terms of evolutionary theory. Another conclusion that evolutionists have come to: some evolutionists are reconsidering the assumption that all life on Earth shares the same basic molecular architecture and biochemistry, and instead examining the possibility of independent evolution and multiple origins of life's different lineages. So we looked at the differences in the AARSs and in the DNA replication and in the lipid biosynthesis pathways. And this is all leading evolutionists to ask the very awkward question, gee, maybe we don't have common descent after all. So here we have the title of this paper, Did DNA Replication Evolve Twice Independently? Here's another paper, quote, altogether, these facts can be seen as an indication that cellular organization has possibly evolved 2 or more times, and that Luca, the last universal common ancestor, may not have existed as a cellular entity. This is a fundamental departure from evolutionary thought and common descent, and it introduces a massive internal contradiction. So remember, last time in false prediction number 1, we talked about how because the DNA code is universal in extant species, evolutionists must say that it was present in the Luca, the last universal common ancestor of all life, and it's a major confirmation of universal common ancestry. They've all got the same DNA code. Now let me go backwards to this here. Altogether, these facts can be seen as an indication that cellular organization has possibly evolved 2 or more times, and that Luca may not have existed as a cellular entity. And this one, did DNA replication evolve twice independently? This is completely at odds with this common descent model and the DNA, the universality of the DNA code. You have a massive contradiction, internal contradiction in evolutionary theory. So we've seen a prediction, we've seen its failure, and we've seen the response to the failure, and it's not pretty. What does this mean in terms of theory virtues? And that's what I've been talking about, that I want to use these failed predictions as a way to evaluate evolutionary theory in terms of theory virtues. And we're looking at these 8 theory virtues: evidential accuracy, causal adequacy, internal consistency, internal coherence, universal coherence, simplicity, durability, and fruitfulness. And as you recall from false prediction number 1, we found that it failed on all but one of these theory virtues. And the one it didn't fail on was internal consistency. On this time around with false prediction number 2, let's look at internal consistency here. The theory's components are not contradictory. Well, on false prediction number 2, we're definitely failing on this third one here, internal consistency. So how does it look across all these 8 theory virtues? Well, it fails on evidential accuracy, it fails on internal consistency, as we just said. It fails on internal coherence, universal coherence, simplicity, durability, and fruitfulness. So another false prediction that has not led to new knowledge or some smoking gun or aha moment. Instead, it isn't pretty. It is the theory is failing across a broad set of important theory virtues. Religion drives science, and it matters.